So let's get started. So the first thing I would like to tell you is how this webinar is going to take place. It's going to take place in free time in 3 moments. The first moment now, I'll take 5 to 7 minutes to present you who is Hyphen, what do we do, and what's new. I guess you all have heard of Hyphen, whether on LinkedIn or somewhere else, but I'll try to take you a bit of the time to introduce you where we are. Then a second moment will be about making a demo of our platform Cloverfield as well as our tools. So I'll take a bit time of that to show you a bit what we can do with our platform that also evolved a lot recently and to show you all those exciting fixtures we have. And then the third time, probably the one that will interest you the most is some practical use case on how to use this digital phenotyping for breeding. And what are the things that I'm allowed to tell you that we're doing with some customers or with researchers. So that's going to be the planning of today. Okay, we are already 4 minutes out, so I'll get started now that I told you what's going to happen. So first about Hyphen. So before Hyphen, I'll introduce myself. I am Alexis Komor. I'm the CEO and founder of Hyphen. Hyphen is a company that was founded more than 10 years ago now, that is working worldwide. We are operating in 18 different countries. Um, we're 30 employees today, so kind of big, already a big organization for phenotyping. Rooted, our R&D and our headquarters are based in France, in Avignon, so south of France. Few words about Hyphen. So 4 years ago, we reached the 20 employees milestone. And we started to really speed up our expansion at worldwide, where we acquired a company that was also doing some drone phenotyping in 2023 named SlantRange, which allowed us to scale a bit our operations in the US. And end of last year, beginning of this year, we acquired the phenotyping division of Aurea, which is allowing us to have a bigger footprint in Netherlands. We have a subsidiary in US. So if you are from US and that you need to work with an American company, we're able to do that. And we'll see. We have different products, and we launched some of them last year that are really exciting, even if it's not fully for breeding. So Hyphen is about really transforming sensor data into agronomic knowledge and decisions for agriculture. So we're not just working for breeding, we're working in many other use cases, but it's always the same thing: is how can I transform a signal into something meaningful for a company? And we mainly work with private companies. We have 5 main products. Namely Phenoscale, Literal, Phenobile, Phenostation, Phenolite. So our specialty is really to process the data at scale. So if you need to process a lot of images at scale, that's where we are really good at. And sometimes we sell a little bit of hardware on top of it to make sure that you're having good data, as we say, garbage in, garbage out. So that's why we do that. Phenoscale is about drone phenotyping. On drones, we're completely signal agnostic, so we can use any type of drone that is available. Today, most of our customers are using DJI. Once again, in US, we're able to operate with other types of drones that are available when you're not allowed to use this type of brand. Literal is a handheld device. I'll present it to you very shortly today, but we'll make a dedicated webinar on that in the coming weeks, months. It's really exciting when you're having small experimental trials below, let's say, 100 to 200 plots, because it allows you to take some real high precision images with less constraints than with drone. Phenomobile, so this is all what is sensor head that we're measuring on sensor head to measure on tractors. Phenostation and Phenolite, which is mainly done for post-harvest assessment or plant phenotyping. And as I said, our job is really to transform all this data online from your data set to some modeling in order to extract traits. We have today more than 70 traits available that we will explore together in this webinar in our glossary. And we can basically cut all those traits into 5 big categories, what we call biomass proxies. So all those things, LAI, above volume, etc. Plant stress, which can be biotic or abiotic stress, disease assessment, and other. Harvest index and quality, which is going to be about looking at the reproductive organs, if that's where you're making your yield, counting the number of spikes, of heads, of capitule, of whatever. Canopy development, which is really about dynamics and how canopy grows, and something that made us a lot of business for those last 10 years is trial quality. Can I really trust the data, like the yield or the other type of data I capture on my trials? So, as I said, we don't only serve seed breeding. Actually, seed breeding is not even 50% of our turnover. It's clearly our biggest area of operations. We don't operate only on that. We also operate a lot on post-harvest assessments, which actually can be, especially in the veg sector, very linked to breeding. Crop protection and nutrition, where I'm really excited about all the things that are happening lately with APPO standards, etc. Agro production, we can think of vertical farming, for example, where we have been involved. And of course, we do still work a lot with research centers and academics in general. And okay, and so that's very shortly who we are. Now let's go onto phenotyping and our platform. What is really important before I go and do the live demo of chlorophyll is that you keep in mind that the big promise today of Hyphen is what we call OTP, OTP for on-time phenotyping. Today, everyone can do phenotyping. I mean, processing data, extracting trait is something that is available to everyone, and everyone is able to do it themselves. The big question is, are you able to do it at a good precision and fast? And that's really the promise on which we're working hard, is how can we process your data in 1 day, in 1 week, in 2 weeks, but make sure that you're having the data during the growing season to be able to take decisions straight away and not wait for a month, 2 months, 6 months, a year, sometimes more, to have your data. So that's really all what I'm going to show you has been put into that. Okay, so that's the end of the first part. Now let's go on to our onto chlorophyll. I hope you can see my browser. So here we are into chlorophyll now. So it's hyphen-chlorophyll.com. If you want to have a demo account, so all what I'm going to show you, you can have access to it. How can you have access to it? By writing to someone from Hyphen, me or my colleagues from the sales team, and asking them for a demo account, and we'll be happy to set you one up. So the first page you can see on chlorophyll is this one, where you're having access to the world map. We'll go more into detail later. And there you can see that you're having those 9 little points where we are having the landing page for each of our products. So this is going to send you to other websites where you can see how our products are being made. By example, if I click on Literal, it does open straight away another website where we describe inside. So what Literal is about, it's the handheld system I described you, and you'll have all the information regarding that. So how it is done, the fact that you're having 2 RGB cameras, 1 near camera, the fact that we're processing the data live on time, etc., etc., etc. You can also just type hyphen-literal.com and you have access to the same information. Same thing goes for Phenomobile, where you can access and discover what Phenomobile is about. Phenoscale, as I told you, it's drones, and I'm going to show you the data now. Phenolite, Phenostation, and Pheno Research. So that's the first thing you have access is to that. Then we have access to the world map, which we're going to go and see more in details after. You'll have access to analytics, campaigns, monitoring, glossary, upload. I'll go straight away into the world map first to show you how it looks like. So there, once you go into world map, you can start to see um, all your trails everywhere. We're going to see and go and see the Melon use case right now. So I go into the Melon and I can see my auto image with my microplot. So I can zoom in to see it better. And when I click on a microplot, I can see um, the image of a microplot. And when I look into the image, I can see the time series of all the flight I did. I can, of course, have access to all my traits that are being here. So here I can see all the traits we computed for these dates. I can decide to not look only on green cover, but on other traits here into my microplots or at other, oops, sorry, or at other dates. So there I'm choosing, by example, to look at how my trail looked in July instead of September. So that's the cool thing. When you want to look at, okay, you're wondering what are the traits and how they look, you have access to a tab that is named Glossary. And the glossary details give you detail of the 70 traits we're having access to. So by example, we're giving you access to some vegetation indices. We describe the vegetation indices here. So that's the simple stuff we're able to do, like NDVI, MCARI, MTCI, etc. We're able to go way further than that by giving you some traits like what is a plot area, how do you compute a plot area, plant lodging, how do we look at plant lodging, and how are we able to see it for 3D point clouds, plant gaps counts, plant gaps distance, plant gaps percentage, plant count. So this is something that we're able to do that works quite well. And each time you have an illustration to show what this trait is about. Physiological stage is also an important one. Are we at flowering or not? How many percentage of flowering are we? How do we calculate this percentage of flowering using some hard case like here in canola? So you can see a very heterogeneous microplot in this case. And how do we give you a score to tell you if it's flowering or not, etc. Organs count, organs area, leaf area index is also something we do through inversion of ProSale, etc., etc., etc. So if you have questions on that, please don't hesitate to, well, first write it in the chat. I'll try to answer it at the end of this presentation. Otherwise, to work on that in a specific call, max plan height, etc. So this is the glossary. So now when I go back to my traits, I can see in my world map that I'm having access to my trails with all the microplots, with all the dates as images, but also with all the dates as here you can see as traits. The second thing you can go and do is to go to the analytics. So we're having today 3 tabs, and there's a 4th tab that is coming up soon. So the first tab is main data exploration. Second tab, and I'll show how we use it, is diversity mapping, which is clearly for breeding. The modality characterization is more for plant protections and plant and product evaluation. And we'll soon have a new tab that is called predictive breeding to allow you to make some predictions through modeling directly within this platform. Why using this platform? Again, and I'm insisting on that, it's on-time phenotyping. So the idea is that you're able to do everything very fast to see if there's some effect and to see if there's some things that are interesting in your plot trails. Once this is validated, you all have some great ideas on how to process this data better, make better predictions for your breeding program, and you will be able to download all this data properly and easily through an API that we call that is here. So it's an API of Cloverfield that is fully documented, that tells you how to use it and how to have access to all your data to be able go further than the analytics I will show you. So when I go to the main data exploration, you can see everything is really fast. So you can see your different dates. You can see your different modalities here. I can select some of them, and what I'm going to see is here at this date, I select some that are all at the same place on the Literary Index. I can go and look at how they look like, and I can see also how they crash. If you're not convinced about the traits we made on the images, so I can see the images and you say, okay, maybe the Literary Index is not the good, maybe I want to look at diseases or something else. You can go to rate the plots, and there we can put you as many scores as you want. So you can really set that and say, okay, this microplot has a scores of 2, quality of, well, it doesn't look very healthy, but this one looks a bit more healthy and with a better score. This one looks really infested and with a very low score, etc. So you can score and save all the information you put there, and of course we'll be able to use this information you use to score to learn together on what you're doing. So that's the first thing. The second thing is when I go back into world map and I say, okay, I'm having, let's now assume that this thing was a nursery, and I say, okay, I want to choose the 10 best plants to go and sample into my field to bring it back into the lab and to characterize. I can make some cost function directly live where I can say, okay, I want to select the plants that are having the green cover that is in those 70 best percent, but I also want some plants that are having important leaf angle, that are having a leaf index that is also significant, a leaf chlorophyll content that is significant. I apply those filters, and I will see the only the plants that I have selected. I can cancel, I think. Wait, how do I go back? And I can see the one that I selected and the rules I put. So this is something that is achievable, and we'll see how we use it in the future. Okay, now if I go into my the other thing I can do is go into my diversity mapping. My diversity mapping is about making PCA, Principal Component Analytics, live into this platform so that I can very quickly be able to assess what is happening. So as you can see, I can choose to put in my PCA all the dates or not. I can choose to put or not some vegetation indices, and I can look then at how it looks like here, my individuals. Of course, I can colorate per attribute, so this is not yet, so I can, by example, colorate by block. So here I'm having 2 blocks, and I can see how my 2 blocks are behaving. But what is really interesting is I can look at the time series of each individuals with its name. So here I'm taking some extremes individuals of on the 3 categories of my PCA, and I can look how they look differently. And so I can see they don't have the same leaves, but I also can see that they are not responsive the same way to disease. Here I can see the first one I choose is a plant that seems to be extremely resistant to disease all along the trail. The last one, the 7459 that is here, seems to be kind of tolerant, or at least has declaration fast, but is not completely dying off that fast. And the last one, the 740, and this one, the center one, seems to be extremely sensitive and have a lot of disease. So if I want to look and select all the plants that are for my breeding purposes or whatever, that are next to the one, my reference cultivar that seems to be more resistant, sorry, this one, I can go and select all of them. I can make a list of variety, and then I can export them to be able to go and pick them into the field. That's the different things you can do with the analytics. Of course, we also have a modetic comparison. I will not show it to you here. It's mainly used with literal, and it will be the main topic of the next webinar Alexandra will do in a couple of time. The last things that are really cool with this tool is first that it's easy for you to upload your data. So if I go to main demo, it's really easy for you to go and upload your data. So you define the data you want, you may create, you just drag and drop your images, and then we take care of all the processing. And when you're having some issues about processing, so for some customer, we're processing up to 1,000 data per year. So with 20 different drone pilots in several countries and in several locations. Well, you want to follow up what are the sites that have been delivered, are the sites that are being processed, what are the sites that are on hold because we don't have all the information to be able to process them. We want to know what are the sites that are having flags. We want to go and read comments. We want to group that, by example, countries. We want to group it by crops, etc., so that the project manager on your end, on the end of the user, is able to follow up all what is happening, and that everything is documented. By example, here we can see that in this plot, we had a problem, a data set incorrect, a plot file missing with infestation, etc. That has been documented with a comment, and of course, we can leave a comment on top of that. So it's really a tool of monitoring that is made to accept that when you collect data, it's messy, there's many people, respecting protocols is not always easy, and we're here to help you. We're here to help you how? By giving you all the information, once again, on time, so that we make sure that you computed, that you acquired the right data, and at the right speed. I showed you all the features now of Chlorophyll. We've seen the upload, we've seen the glossary, we've seen the monitoring, we've seen the analytics and the world maps. Now let's go and see how all those data are being used. I'm going to present you very quickly 3 main use cases that we're using for breeding purposes, 2 with INRAE, which are a bit vertical, and 1 with a company that happily allowed us to share some of the results we're having together on strawberries. We'll see that the second and third use case is the only thing I'm allowed to say on what we're doing in predictive breeding, especially using phenomic predictions. And so we'll try to go along those lines too. So regarding the strawberry use case and the kinds of things we're able to do, their problem is in strawberry that you're working with an octoploid, you're having nurseries where in their case you're having 10,000 plants. You can have, of course, way more. And they wanted to select the plant that they crossed. Basically, they have 2% cut, so they wanted to select 250 plants out of 11,000 that are having a desirable traits on precocity, biovolume, number of fruits, and size of the flower. And as you can see, when you put a strawberry plant on the ground, it's not going to survive very long, so you want to make your drone acquisition. We made 6. And you want then to go and get your plants extremely quickly to continue growing them into greenhouses, and then to be able to advance your breeding program. And so the dynamic filtering I showed you was designed to be able to do that, was able to give the tools to the breeders to select the best plants in their nurseries really quickly. And outside of 11,000 plants, so it's starting to be a significant chunk of data where they had access to all the pictures. And we compared together a bit how the sampling happened between the tool of Chlorophyll, so those are the green points, compared to the breeder that went to sample his own plants that are the white, red, and blue points, where there are 3 different colors because he went to get those plants and 3 different times during the growing season. So what you're saying as a graph is a diversity map. Huh? So we did a PCA as I demonstrated to you on chlorophyll with individuals, and roughly the first axis of this PCA is linked to vigor. So we can see an individual here on this side of the graph that is having a very low vigor, and on the other side of the graph that is having a really high vigor. And you can see that in the other axis, it's basically number of flowers, number of fruit. It's more or less linked to that. It's a PCA, so it's never completely linked. So you can see the number 3 where you're having very little flowers and fruits, and the number 4 where you're having a lot of flowers. And what we can see is that we didn't select it at all the same way with chlorophyll than the breeder. And the main reason is not that the breeder is taking bad decisions. He's generally choosing very well his plants. It's more that when he goes to the field, he doesn't have a time series in his head. Normal, there's 11,000 plants. And so when you are able to make decisions, your traits on a time series, you're able to select it's easier for you to select the one that have a high precocity, as I told you that was a very important trait, as well as a strong early vigor through biovolume. So that's why probably we have been more accurate in selecting the right plants um, on our 250 plants. Of course, what they do with these plants and all that will not be discussed today. And you can think of actually making this type of diversity map using AI in a more advanced way. So there is what we're doing, where we're trying to go into the latent space. Huh? So to go to the latent space is to use autoencoder, and I'll speak of that a little bit just after the slides of how we do that, where we can see that when you autoencode two plants that are very similar, where you're having very similar vectors, and when you encode fruit plants that are a bit dissimilar, you're having some dissimilar encoding features. And of course, you can see that the number 4 here is kind of similar to the number 1 and 2. And when you plot that with a methodology that we call TSNE, you can also see the diversity of your flowers, of your plants, sorry. And you can imagine when you're able to spread your diversity like that you would like to select some plants of high precocity, by example, for Spain here, of lower precocity for the German market, that would be here. And you could try to put some reference markets plants here in this graph and help selecting the ones that are similar but with different characteristics. Of course, that's what we can do with drones at the first level. I told you we have some new tools that are Phenolite and Phenostation that allows you to measure also the post-harvest quality. And all the game of the work we're doing is how can I predict when I measure the plants with the drone, how would look the fruits. And of course, actually, we want to go further. How, when I cross two parents, can I predict the fruit of the next generation? So we can choose who to cross with whom and who will have the most chance to make the best next cultivar. Just using predictors, so just using images and not using genetic information. That comes, and that's a nice transition to a quick focus before showing you the two other use case about phenomic prediction. What is phenomic prediction about? Phenomic prediction is about predicting predicting how a trait based a trait or a breeding value or a different series of traits based on sensor data that are can be captured on something related to my trait. So by example, I want to measure some sensor data on the parents to predict the hybrid or to predict the next generation or to do things like that. Trait phenotyping, on the other hand side, is to transform my signal through physical means into something that I can validate it against ground truth to some traits that I really know are linked to some physical characteristic of my plant. So in one case, it's about making modeling and predictions. In the other case, it's about making modeling, but more into a more traditional approach to kind of replace ground truthing, or at least ground measurement, because the ground saying that it's ground truth is not always ground data is not always truthful. So it's a concept that has been, I guess people are thinking of it for a while, but the first paper that is really speaking about it that I found in the literature is in 2018 by Renaud Vincent. And what he did was using NIRS data. So he was using a spectrometer and using the spectrometer data as predictors to be able to predict in the materials something. The vision that we have at Hyphen is to say that, okay, a spectrometer is cool, but it's just one dimension of the dimensions of phenotyping. Yeah? So the wavelength is always goes from UV or even X-rays to radar bands. When I use a spectrometer like that, I go from 400 nanometers to 250 nanometers. But I can also make image. I can also make 3D images that are three other dimensions. Depending on the angle in which I make my images, I'm also going to have different signals, and the time is also a very important dimension of phenotyping. The good way to see time is, by example, fluorescence. Depending on when I send my source and when I measure the stimulation of a plant, I will not have the same signal when it's the same plant. All the six dimensions are really important and could be used as predictors, data that are collected in the six dimensions, to make phenotypic predictions. Of course, all our tools allow you to sample the six dimensions, and something that we are working hard since always. And how can I transform all those data in six dimensions into predictors? Well, I have to synthesize them, and there's two approaches to synthesize them. Either what I call the signal and trait approach. So there's a nice paper from Seth Murray and Al Pradak from Texas A&M speaking about it, where they used actually VIS, vegetation indices, in a temporal way. So they use the temporal information with vegetation indices to be able to make some predictions, some phenotypic predictions. You can also play with autoencoders, so use the latent space, as what I showed you on strawberries, to try to synthesize all this information. So that's AI. It's basically a bit like ChatGPT is working. So we're using transformers, and it's really the same type of mindset as GPTs. And so concretely, how does it work? I'm going to show you a first use case that is simple, which we have been developing in the framework of ELEX. ELEX is a European project about sunflower, with all the main research center working on sunflowers and also the breeding company, where we had our task was to predict seed humidity. And what we did was a pipeline where we extracted from this pipeline each microplot, each sunflower head, and from each sunflower head, we went into the latent space to synthesize it, and then we correlated it with seed humidity. We did that in 2023, with whom we use the same data set for training and, of course, for validation, of splitting it, but we had a great relationship. We validated it just by training it in 2023 data sets. In 2024 in France and Spain, with almost as good correlation than RMSE. So that was really cool. And as you can see with RGB, you don't have direct access to seed humidity, but as seed humidity is highly linked to its maturity, to the physiological stage and the color of the sunflower head, we're able to find some correlation using AI that seems to be repeatable on time and in different locations. And you can see very high is the estimation and food dates. So you can this way follow up the drying of the seeds and the speed of maturity that is arriving. The last use case I would like to show you is some work that we have been doing, that mainly INRAE has been doing, on which we have been in support, where we try to compare genomic predictions with phenomic predictions, at the same time using NIRS data and drone data, trait or latent space. So for that, we did, it's an experiment that is complex, where we had some incomplete factorial designs, where they knew how the hybrids were behaving, and we measured only the parental lines, Flint and Dents. And basically, we did 6 dates, and we managed to extract 33 traits per date. Those traits were 8 vegetation indices, a procell inversion with LA, chlorophyll, height by volume, but also some segmentation of green cover, greenness, plankton, presence of panicules, etc. On the other hand side, we tried to bring all this data into latent space by removing the environmental effect. So we removed all what was linked to the soil and to the and also, so that's what we did first, removing the soil, and then trying to take free plants and not just all the microplots, to be able also to have just the plants that are behaving as a canopy, to try also to remove this environmental effect, that is that plants are tending to grow faster when they have no neighbors. And what we saw so far, so it's ongoing work, is that when you're using all your genetic diversity to train your model and then to validate it, so that means I'm using some individuals of each family. Genomic prediction, everything works very well, and genomic prediction is a bit superior. When I'm using some new families that I want to predict how they're going to behave with a training data set of other families, well, genomic predictions is not that predictive, and drawn with traits is what is the most predictive in this experiment. So it means that you're having valuable information that is probably disjoint from genomic predictions. Most breeders, I think, that tried this thing are saying that when you combine the data, and there's a nice paper from CIMIT saying it, when you combine genomic and phenotypic data, that's where you're the most predictive, which makes sense. Voilà, so these are the types of things we're doing. We're having a lot of idea. I see I'm a bit late on time. I took more time than I expected to present all of that. But basically, we have a lot of idea on how to improve that, how to try to separate the environmental features from the genetic features. There's lots of really exciting bibliography that suggests that there's lots of really exciting things to do. Whether on AI to try to remove the environmental effect that is not really convincing yet, whether on traits where we can do more traits, we can try to test the predictive factors of each trait category to try to be cheaper, and we can also try to make some user interface UA to test those predictors for all what you want to do. And that's what is going to be the tab predictive breedings in chlorophyll that we're currently developing to make you able to do some predictive breeding really easily on our platform live and very quickly, knowing that then you can have some better models later on if those predictors are meaningful for your task and for your breeding program. So maybe that's the last thing I would like to speak, and of course I could go further, but maybe not for today. All of that is about phenotypic prediction pipeline now, where actually you need to define your targeted traits. So when you're in vegetables, there are generally a lot of traits that you're focusing depending on your markets. When you're in field field crops, generally you're having yield, and other traits are of less relevance, often, not always. All of that is going to allow you to define what are the predictors you need to have, and there's no magic. There's some papers saying that when you're trying to look at what there's in roots, like in potatoes, the starch content and stuff like that are not very well linked to the aerial signal you're having with drones of leaves. So the sensor data needs to be as much, is going to have a huge influence on whether this method works as not, so whether the predictors are meaningful, as well as the processing tech. I showed you those 3 different processing tech we use: image to traits. So that was what I showed you with the maize. Image to latent space. This is what I showed you on sunflower. and time lapse to latent space. This is the diversity mapping we did in strawberries, for example. That allows you either to do a mapping diversity to be able to look and select the crops that are similar to a target, either to make predictions when you have a training data set to be able to train and to make your predictions. Please remember that phenotypic predictions is not genomic predictions, and it's both are really good, important, complementary, etc., etc., etc. I don't want to get enemy with no one. And yeah, and that's it. And here comes the questions. I'll share sharing right now. I'll go back into the main page and try to go and answer to your questions. For the one that had just planned 45 minutes, I'm thanking you very much for having listening to me. Please get in touch with me or with my team if you want to have additional information.